After reading this module, you would be able to understand the following mechanisms of enzyme action. Enzymes catalyze biochemical reactions much the same way as chemical reactions are catalyzed by inorganic catalysts. Due to the large number of amino acid residues present at the substrate binding site, contributing to a large number of diverse functional groups, enzymes can use a variety of catalytic mechanisms. In multi-substrate reactions, the large substrate binding site can accommodate all the substrates in close proximity to each other to facilitate transfer of groups. In some enzymes which have reached catalytic perfection, every substrate colliding with the enzyme is steered into its active site, leading to a reaction. Let me introduce you to this module. One of the amazing features of enzyme action is that they are able to increase reaction rates to over 10 to the power 14-fold. The reactions catalyzed use mechanisms similar to chemical catalysis like acid-base catalysis and others. However, since enzymes possess a variety of functional groups at their substrate binding site, a number of mechanisms can be used simultaneously, unlike in inorganic catalysis. For example, in a routine chemical reaction, the reaction can be catalyzed either by acid catalysis or base catalysis, never both. However, in the case of enzymes, basic and acidic groups provided by different amino acid residues can perform a concerted acid-base catalysis, leading to a faster formation of product. Other mechanisms such as distortion of the substrate or substrates to convert it into a high energy transition state, steering multi-substrates within proximity for interaction, and providing functional groups for transient covalent bridges help to increase reaction rates. Let us now look in detail at different mechanisms of enzyme action. Enzymes are large proteins which fold into unique three-dimensional structures depending on their amino acid sequence. Within the enzyme structure is usually a niche or pocket wherein the substrate or substrates bind. Although enzymes are water soluble and cells within which they act are at least 90% aqueous, the substrate binding site can be inaccessible to water due to hydrophobic amino acids present around the active site, thereby allowing reactions without water such as electrostatic catalysis to occur. Water, being highly polar, can otherwise prevent certain reactions from occurring. This is the same as reactions occurring in hydrophobic solvents. Enzymes, therefore, can provide a biphasic medium to enable certain mechanisms to occur, namely mechanisms which operate in a non-polar environment. One of the major ways by which catalysis is performed by most catalysts, inorganic, organic or enzyme, are acid-base catalysis. In acid catalysis, a Bronsted acid donates a proton to a substrate, allowing it to attain a high energy transition state which facilitates its conversion to product. In the case of a base catalyzed reaction, a Bronsted base extracts a proton from the substrate, leading to the formation of a transition state intermediate. Let us look at the reaction of a base catalyzed ester hydrolysis. The ester, indicated as R carboxylate group and O-methyl, undergoes first, in the presence of a OH minus, the addition across the carbonyl bond, giving rise to O-methyl structure. In the next reaction, O-methyl is removed and there is the formation of the acid. In an acid catalyzed ester hydrolysis, the ester interacts with an H plus, which is fast, which adds to the carbonyl group, and then next in the slower step you have the removal of the proton with the release of an acid and a alcohol. Let me say this again. Acid or base acts as a catalyst on an ester to release the products alcohol and acid. Remember that either the reaction can be acid catalyzed or it can be base catalyzed, but never both. Similarly, in the case of mutarotation, a phenomenon observed in glucose wherein alpha D glucose and beta D glucose can interconvert with changes in optical rotation through the formation of a linear complex. Acid or base catalyzes the interconversion as indicated in the figure. If one has dissolved the beta isomer, the cyclic structure in the presence of a proton undergoes an opening up wherein you get transiently a linear structure. When the proton leaves, then there is a ring closure to form the cyclic hemiacetal again, except that you can have the formation of the alpha isomer, or in other words, if the beta D glucose is dissolved in water and exposed to acid, then it can convert to either the alpha or the beta, and this is because there is a ring opening due to the protonation, and again when the proton leaves there is a ring closure. Therefore, both the anomers are then formed finally in solution. The interconversion between alpha D glucose and beta D glucose is enhanced in rate by presence of both acid and base, as is indicated in the next figure. Here, instead of the acid, a base is involved in opening up the ring, and you can easily observe how the same process, the cyclic structure opening up to give you a linear structure, and then the linear structure closing again to give the cyclic structure, thereby having the OH group or the hemiacetal group moving either over the plane of the sugar ring, and this leads to the interconversion of the anomers of glucose. If the acid and base groups, instead of being separate, are on the same molecule, such as alpha pyridone as indicated in the figure, the reaction is catalyzed with an enhanced rate of 7,000-fold. This indicates that the concerted acid-base catalysis is more efficient, and in the case of enzymes, this is easily provided. Let us look at an example which illustrates this concerted acid-base catalysis, namely the reaction of a typical enzyme ribonuclease A, which catalyzes the concerted acid-base catalysis of a phosphodiester bond in RNA molecules as indicated in the figure. The enzyme RNase A, which is active in the slightly alkaline pH range, has two critical histidine residues, histidine 119, which acts as an acid, and histidine 12, which acts as a base. Please note that the histidine imidazole structure which has a pKf around 6, can around neutral or slightly alkaline pH, can act as both a proton donor as well as a proton acceptor. The simultaneous activity of both histidines causes the breakage of the phosphodiester bond, as is clearly observed in the figure. The cyclic phosphate released is acted on by water in a reversal of the same mechanism. Please note that histidine 12, which lies on one side of the phosphoester diester bond which has to be hydrolyzed, and histidine 119 is on the other side. Initially, there is a reaction wherein the histidine 12 extracts the proton from the base ribose group on position OH group on position 2, thereby facilitating the formation of cyclic phosphodiester bond. Now let us look at another form of catalysis, namely covalent catalysis. In covalent catalysis, a transient covalent intermediate is formed between the catalyst and substrate, facilitating electron induction or repulsion through the covalent bridge. Facilitating electron induction or repulsion through the covalent bridge. Let us look at the reaction of the decarboxylation of acetoacetate by covalent catalysis indicated in the figure. The reaction is catalyzed by primary amines by the formation of a transient Schiff's base between the primary amino group of the catalyst and carbonyl carbon of the aceto group. In an analogous reaction catalyzed by the enzyme transaminase as shown in the figure, the coenzyme pyridoxal phosphate forms a Schiff's base with the keto acid. The Schiff's base, which forms a bridge between the catalyst and also the substrate, allows protons or electrons to move through. In the reaction catalyzed by the enzyme transaminase between two substrates, an amino acid and a keto acid, wherein the two are interconverted, a transient Schiff's base is formed between the coenzyme and amino acid. The resulting pyridoxamine form of the coenzyme is released along with the keto acid. The pyridoxamine then forms a Schiff's base with the other keto acid, transferring the amino group to it. Evidence for the formation of the Schiff's base is provided by capturing the complex using the reducing agent sodium borohydride. Another form of catalytic mechanism is electrostatic catalysis. The high dielectric constant of water and therefore enzyme catalyzed reactions in aqueous reactions would ordinarily prevent many reactions between substrates which involve electrostatic interactions. In certain enzyme catalyzed mechanisms, water is precluded in the presence of substrate to facilitate greater electrostatic interactions as in an organic solvents. Many pKa's of amino acid side chains vary by several units in the absence of water, thereby increasing electrostatic interactions. There is evidence that such amino acid residues can guide polar substrates to the enzyme active site, increasing enzyme reaction rates. In a previously explained reaction mechanism involving chymotrypsin, and if one observes the figure given, decrease in pKa of serine hydroxyl group in proximity to a histidine is clearly indicated in the figure. In the hydrolysis of a peptide bond by the serine proteases, the serine OH group, which has a pKa of over 10, is able to release a proton to the nearby histidine imidazole ring, facilitating the reaction. This is possible only because water is prevented from entering the active site, allowing the serine OH to develop a lower pKa in the proximity of a nearby base, namely histidine. Another useful and important way of enzyme catalysis is metal ion catalysis. Many enzymes require metal ions for catalysis. In some cases, the metal ion is part of the three-dimensional structure and is a transition metal like Fe2+, Fe3+, copper 2+, or manganese 2+. Such enzymes are called metalloenzymes. The figure represented is the structure or the three-dimensional structure of carboxypeptidase A with a centrally located zinc 2+ ion. In several enzymes, metals ions participate directly or indirectly in catalysis by loosely binding the enzyme, and such enzymes are called metal activated enzymes. These metal ions contribute to the catalytic mechanism in the following way. 1. Strongly negative substrates such as nucleotide phosphates are bound with the metal ions to prevent repulsion on approach to the enzyme. 2. Copper and iron as well as other transition state metals catalyze oxidoreduction of substrates due to reversible changes in the metal ion's oxidation state. 3. In some enzymes, the substrate is oriented for proper reaction by binding to the metal ion. Look at the mechanism as given in the figure for the action of carbonic anhydrase. The enzyme carbonic anhydrase catalyzes the solubilization of the gas carbon dioxide to form bicarbonate. In the mechanism of action of carbonic anhydrase, the zinc ion of the enzyme is bound with a molecule of water. This orients the incoming carbon dioxide molecule to bind in a way facilitating the formation of bicarbonate ion with the release of a proton from the water molecule. In enzyme mechanisms, proximity and orientation effects play an important role. For any reaction to occur, reactants must be properly oriented spatially. The enzyme substrate binding site enables substrates to bind proximal to each other with the correct orientation to enable reactions such as group transfer as illustrated in the figure. Let us look at the figure which shows the proximity effects in substrate level phosphorylation by pyruvate kinase. The enzyme pyruvate kinase catalyzes the transfer of a phosphate group from a glycolytic intermediate phosphoenol pyruvate to adenosine diphosphate to form adenosine triphosphate and release pyruvate. The binding of PEP or phosphoenol pyruvate and ADP or adenosine diphosphate in close proximity and correct orientation is possible due to the structure of the enzyme substrate binding sites as illustrated. Hence, preferentially ATP is formed rather than the phosphate group getting transferred to water. Let me summarize what you have learnt in this module. Enzymes use a variety of catalytic mechanisms to enhance reaction rates. Mechanisms used by enzymes include acid-base catalysis, covalent and electrostatic catalysis. Enzymes provide suitable substrate binding sites to correctly orient and bind substrates proximal to each other for reaction to occur specifically. Enzymes can bind to metal ions tightly or loosely to form the correct three-dimensional structure and thereby promote catalysis respectively. Since several mechanisms are simultaneously occurring, enzymes can catalyze reactions at enhanced rates.